He'll be in the video.
I know. Well, I'm just in it. Good evening, everybody, and welcome to the next in our fall of 2017 idea talk series to be differentiated from data idea seminars or our pirates meetings or Tetherless World TWED talks. Tonight, Hannah de los Santos is going to talk about her. Now, it's your PhD work, is it actually?
Yeah. Eventually.
in the circadian rhythms, detecting circadian rhythms. Just a couple of logistical things. Hannah's going to go through a presentation, but you're also welcome questions.
Yeah.
And we just need to wrap it up by 7. This is being recorded and streamed. Just keep that in mind, with your questions and discussion. And for any of the Tetherless World people in the house, remember to sign up for TWED lightning talks at the end of the term. And actually, idea people are welcome for those too. So get out of your way and thank you, Hannah.
Yeah, sure. Yay. So as he said, I'm Hannah de los Santos. I'm going to present ECHO, Finding Circadian Rhythms Using Extended Harmonic Oscillators. Long title. But yes, I'm Hannah, and my advisor is Kristen Bennett, and I work with Emily Collins and Jennifer Hurley, Professor Jennifer Hurley in the biology department. So just a quick overview of what we're going to do. I'm just going to go through a quick introduction about what are circadian rhythms, how do we identify them, some methods, actually how we actually get these according to my method, some results for Neurospora crassa, which I'll talk about in a little bit, our R package and our Shiny application, just its features and stuff, and then a live demo of that. So, as I said, introduction. What are circadian rhythms? So essentially, they're 24-hour endogenous cycles, which means they persist over time. And they're reinforced by external cues such as light, but they'll continue anyway. So if we put you in like a cave, you'd be really sad, but you'd still wake up and go to sleep. So it's very essential to daily function, and interrupting the cycle can lead to a lot of health problems like cancer and diabetes. So we kind of want to know what these are in order to mitigate those factors. And so the way we observe for circadian rhythms, we look at specific organisms, including Neurospora crassa, which is a sort of bread mold. It's the model clock organism, and it has a clock very similar to our mammalian clock. It has about a 22.5-hour rhythm. Mice are also animals and therefore very similar to us. They have a 24-hour clock. There's also Drosophila melanogaster, which is basically just a fruit fly. And it's actually the featured organisms in this year's Nobel Prize-winning work. They identified and characterized the genes comprising the core clock mechanism in Drosophila. And so for most of this talk, I'm going to focus on Neurospora crassa, just because that's data we've been working with for a while. Yes. So I'll talk about that a little bit. So our data, in terms of that, we have 24 time points between 2 hours and 48 hours with a 2-hour resolution, about 9,000 genes and 3 replicates. And there are 6 missing time points, which can confuse things a little bit, but anyway. So what circadian rhythms actually look like in genes, they follow sinusoidal curves. So existing methodologies like JTK cycle and eJTK cycle, which I'll talk about a little bit later in comparisons, they compare experimental data to reference cosine curves, but they use fixed amplitude models. So that means they can get amplitudes that look a lot like this, but they can't really get amplitudes that look a lot like this, where amplitude decreases over time. So in reality, as I said, they might be damped or even driven over time. So fixed amplitude models might miss genes with these patterns. So on the right, you can see a luciferase data figure for Freak. Freak is one of the core clock mechanisms in Neurospora. And basically what they did in that experiment was they essentially tagged Freak and watched it glow and darken and glow and darken over time. And experimental conditions for that, they essentially didn't give any more light. They didn't give any more food. So as you would expect, they would die over time. So their expression also goes down over time. And as you can see, things like that might miss it. So how do we actually identify these? We use extended harmonic oscillators. Very exciting. So basically, this is the basic harmonic oscillator equation that you know from physics a very long time ago. And it has all the standard parameters, so like amplitude and frequency and time, etc. But what we care the most about is gamma, this forcing coefficient. It actually captures the changes in amplitude over time. If it has a positive value, it indicates damping, and a negative value indicates driving. So in order to get these parameters, we use nonlinear least squares, which basically just minimizes the residuals, which is basically the difference between the experimental data and basically just uses a least squared, hence the difference in the square. So we find the local solution using the NLS-LM algorithm, the INPACT-LM package in R. So as I have alluded to, we only find local solutions, so starting points are very, very important. So we choose these starting points with heuristics based on the data, such as the starting amplitude as the max minus the mean, the equilibrium shift is just the mean of the data set, things like that. So for replicates, it's slightly different. So to utilize replicate data, we have to add weights in order to make it an unbiased estimator. There's proofs and stuff like that, but I won't go through that. But so we add weights that are inversely proportional to the variance at each time point, so that essentially we can trust points with lower variance more than ones with higher variance. Yes.
Actually, so if you go back to the last slide,
yes.
I was curious. So does having a good heuristic for starting points, will that affect only how long it takes the algorithm to converge, or will it actually converge to something that's biologically impossible?
I think it would definitely also converge to something that is biologically impossible. So I guess, like, drawing. This eraser. Oh, this is awesome. So I guess you can imagine if I started. So if my rhythm looks sort of like this, a little bit damped over time, and I start with something maybe like this, it might just converge to something that looks like this, right? Which is nothing like the actual thing, but it does minimize stuff. But if I choose something kind of close, like this, it'll probably converge to something close to what the actual rhythm looks like. So it is really important to choose those starting points, and also choosing points close will make it go faster. Does that answer your question? So yes. Yes, so continuing on about that, about the variance and the inverse of it. Basically, what it means when we add the inverse of the variance is we can trust points like this more than we can trust points like this, which kind of makes sense intuitively. We also have some pre-processing steps. One of them is smoothing. So Neurospora and just in general expression data is very noisy. Biological systems are noisy. People are noisy. So we want to smooth that data a little bit, and we use a 3-point window. And we can use unweighted smoothing, which is just basically 1, 1, 1, just smooth everything equally. Or we can use weighted smoothing, where we basically multiply the middle point by 2, and basically that kind of retains the actual expression a little bit. So you can see the red is the unweighted and the green is the weighted. So as you can see, the green kind of retains some of that actual fit, whereas the red kind of just dampens it out.
So you're using both before and after so if you're trying to predict X, you're doing X minus 1 and X plus 1.
Yes, yeah, exactly. So yeah, so it's like a 3-point. We also want to remove unexpressed genes. So things that are clearly not oscillatory at all, we really don't want to look at them anyway. And so these things include constant expressions, basically where there's no change over the entire time course, or less than 70% expression, which is expressions less than 70% of the time course are non-zero. So we have constant expressions like this, clearly not oscillatory, and things like this, where like 3 out of like 16 time points have a non-zero value. That probably doesn't mean much. 70% is just the industry standard, industry biology standard. So how do we actually determine what is damped and what is driven? So we actually look at our forcing coefficient, our gamma value. And we can just look at these different categories to get what is driven, harmonic, damped, etc. Yes?
Just kind of a technicality, but a driven oscillator just means that it has like the original equation that you solved to get the solution that you're fitting. You have a driving force.
Yeah.
So driven doesn't have it doesn't have anything to do with gamma, just like in the terminology. If you have an oscillator that's getting faster as you go forward, you just say that gamma is negative in general. Yeah.
Yeah. Yeah.
It's just terminology, but
yeah.
Yeah.
Yeah. Cool. Yes. Thanks, Alex. We also don't consider genes to be circadian if they have gamma values of greater than 0.1 or less than negative 0.1. And they can be, we consider these to be over-damped or over-driven. So essentially genes that start off really high and then go to really, really low, that really doesn't mean anything. When we used to fit things that only had like one peak right at the beginning, they would fit a lot like that. They would be circadian, but they wouldn't really be circadian because they just start off high and then go very low oscillations. So we decided to cut off those. For Neurospora, with compatibility with JTK cycle, since JTK cycle only generates a bunch of reference cosine curves, you have to specify the range that you are looking at. So we wanted some compatibility, so we also said, hey, let's just look at 20 to 26. And we also restricted the period of the range during runs to that period. So we actually have our fits now, but how do we actually determine whether these fits are good or not? We use a p-value. So what we do is we use Kendall's tau rank correlation coefficient. Very long, but very easy. So basically it determines the concordance between two series of data, or how well they go together. So basically the numerator is the sum that very concordantly they go together, versus minus the ones that go discordantly or they go apart. The denominator just normalizes it to be between -1 and 1. Pretty simple. And then once we have Kendall's tau, we can determine the p-value. For single replicate systems, we use just a normal approximation. For multiple replicate systems, which we have found to be a little bit more complicated, we use the exact distribution. And then we adjust for multiple hypothesis testing using Benjamin-Hochberg, which is a less stringent thing, and Benjamin-Uccelli, which is a little bit more stringent. So moving on to the overall results for Neurospora. So also all of these figures were generated using the app, which I will show you later. So in terms of using the most stringent cutoff, we found that about 200 didn't converge. About 250 didn't even start because they just had too much noise. 0 were unexpressed. 0 had no deviation. And then we identified about 1,600 circadian genes, and about 700 of those were unique to our method. They were not identified by JTK. Of those, we identified 800 as damped, 500 as driven, and about 200 as harmonic. I did not include overdamped or driven genes in this circadian calculation, but you could, but not recommended. And then for JTK cycle, they got about 1,000 and about 200 unique. I looked into why there were 200 unique genes that we missed, and it's because they fell slightly out of our what we considered overdamped and overdriven. So they fell into those categories. Once you include those, that goes down to about less than 100. So that's something.
Can you explain what you mean by unique there? Unique relative to what?
So I said that this was unique and JTK cycle did not, and vice versa. Unique.
So you said,
like, so Echo identified this gene as circadian.
Okay.
And JTK said it was not circadian.
Oh, I see.
Yeah. Yeah. Which is, it's a lot easier to see if you use this a Venn diagram. Very simple. Where you can see the overlap and then the not overlap. So
Which one's right?
Yes.
Does anyone really know?
Can you like look at it? You know, because you can see the picture, right? You're running this algorithm just without looking at it, but you can just take samples to see.
Exactly.
Okay.
Yeah, we can totally look at specific genes, which I will show in my demo. So yeah, good.
Good straight man artist.
So
Yeah.
the genes that were measured in the data sets you looked at, were those genes measured because domain experts thought they had a good chance of being circadian?
No.
Or so these were just genes that are easily measured? And so
Yeah,
the other
yeah.
genes that
Actually,
can't be-
what was the assay?
So what they did was, and I was going to talk about it later, but I can talk about it now. So what they did was, for Neurospora specifically, is that they made 24 time points times 3 clones of Neurospora, and at every time point they took away 3 of those and measured using RNA-seq to get the expression levels at that point. So it's kind of a destructive method. So yes, that's kind of what they
So
did.
they were, so basically they weren't. Right, okay. So
Yeah.
That will be comprehensive.
Yeah, exactly.
Summarized.
Yes.
And because they do it that way, which of the 3 gets assigned to which replicate is completely arbitrary.
Yes,
Right.
exactly.
So-
Yeah. So it's kind of just like labels mean nothing, really.
So you could really make a whole bunch of different replicates out of them.
Yeah, exactly.
You could make the replicates in different ways.
Yeah, exactly. I could switch 1 and 2. I could switch 3 and 2, whatever.
So of the 203, you said 100-ish were
Were-
actually what you would consider overdamped or underdamped. Just
Yeah,
Yeah.
exactly.
But of yours, of those 735, you know those are not,
Overdamped or overdriven?
Right.
Yes, yes, yes.
as well as the 877 that are overlapping.
Yeah, so we agree for 800 about. Yes.
And those 800 were neither damped or driven.
No, they can also include damped or driven genes. So as long as it's close, so JTK cycle includes harmonic things, but as long as it's close enough within a p-value,
Okay.
it will say yes.
So it'll
So
get a certain amount of damping.
Yeah.
Driving, but not much,
yeah, exactly.
especially compared to X.
Yeah, exactly.
Okay.
And what's nice about Echo is it also allows for the knowledge that it is damped or driven, which actually it gives more information to biologists when they look at what functions things do.
So JTK cycle doesn't actually return a gamma
No.
value to you.
Yeah, no, because it just does fixed amplitude. So it's more information. Yeah, yes. So if we also look at just density plots of the forcing coefficient and next period, we can see what I kind of said before, that a lot are damped, so we have a lot of positive values, not as many driven. But there is like a peak at the center, which we'd expect for harmonic genes. For the period, since I stopped the oscillations, or I didn't look for periods outside 20 and 26, you can see that it kind of bunches up. If I had let it go, it would probably go out a lot more. And we've definitely seen that if I do that. And we also see a little peak around 22.5, which is what you would expect since we're looking at Neurospora. So I'm going to talk about the package and application a little bit.
Actually, on that distribution, do you include those outlier period?
No. So these are also, oh, I forgot to mention this, but these are also only the ones that we identified. So no, it doesn't include the damp or the overdamped and overdriven.
Well, no, I'm talking about the periodicity. So since you know that the period in this organism is about 22.5, the further away you get from it, the less likely it is.
Yes,
to be. So
exactly.
Do you take that into account when you're
No.
predicting the genes that
No.
are relevant to this?
Yeah, no, not at all. It's something that you can do also because you would probably see, like, well, we've definitely seen. If you don't actually restrict this period to be between 20 and 26, you get essentially just what you would expect. It starts going down.
Yeah.
And then you get a peak around 22.5. And it's just because we stopped it here just for compatibility. that it does that. Did I answer your question? I don't know
think
if
so.
I did. Okay.
I think that like seeing the peak,
Mhm.
yeah.
Yes.
It's useful information to predict on.
Yeah. And we also really don't know much about, it's essentially, what we're saying is you can put whatever data you want about your organism. You don't really need to know at that much. I don't need to know that I have a rhythm of 23.72, etc. I just put it in and figure it out. So moving on to talking about the package and the application. We're going to develop an R package. So this allows anyone to use the functionality or program in a very straightforward way without having to code too much, which is nice. And it's also very easily accessible to anyone using R. Just if you like, say, install packages. Granted, that's not on right now, but it totally will be in maybe a week. We also developed a Shiny app, which is an interface for users to find circadian rhythms with no coding at all. Very advantageous to biologists who don't like coding. It also allows for analysis of results, and it's very easy to run for anyone who's downloaded R and RStudio. And it essentially is broken down into two pieces, finding rhythms and visualizing rhythms. So in terms of the finding rhythms part, it looks a little bit like this. So you'll upload data and specify your data set information, and then you can run echo and JDK cycle if you want. And then you can download the results as a CSV, and then the R data file for visualization. So you'd upload that R data file that you got from running, and then you'd get a simple method to explore your data results. You can look through summaries and gene lists, and you can also visualize the data in several ways, including Venn diagrams and heat maps and gene expression plots, and also those parameter density graphs, so all of those graphs that I generated before were from this app. I didn't have to do any coding at all. Well, I did originally, but yes. So I'm going to switch to the demo. So I'm going to do that. Not acknowledgement. Well, there are these people, we want to acknowledge them. Yes. Okay, so I will switch to. If you have any questions, you can ask them now while I am switching things.
Do you have any heuristics to try to avoid bad local minimizers, like some sort of like simulated annealing type thing where you intentionally take bad steps once in a while?
No, because I don't really control how, I just basically use someone else's algorithm to get to minimums, so I don't really.
Do you know if like the minimizers you've gotten to are about as good as you can expect, or like do you think that maybe there's better fits in the parameter space that you just haven't been able to explore?
I believe that there would probably be even better parameters than I get, but since I'm trying to do it in like a very high throughput way, I can't do that. I can't like look at different fits.
I guess what comes down to just analyzing the smoothness of the objective function via some sort of simulation.
Yeah.
I'm fitting functions very similar to this, and like the initial guesses are really important.
Yeah, so this kind of takes us in a different direction, so you can put me off until after you've done the demo. But so right now, the samples you're getting, the biologists have determined essentially the sampling rate and what you get. But really, in a lot of the kind of data we'd be interested in, some of your data, things like that, you may be looking at a stiff kind of distribution where any given sampling rate can miss key minima, or you could be looking at something fairly random. So I'm just going to cue that up now and later. Have you thought about what to do about, when we get to data that doesn't have that kind of regular sample? Finding damped oscillation, and that would be incredibly powerful.
Exactly. I will definitely talk about that when you ask about it. Okay, so I'm just gonna, just to show what the data looks like in Excel. We want our data to be in a very specific format. It's just easier to assume it that way. Oh, I should. Basically, we want the first column to just be labels, and then we want time points ordered by replicates. It's just easier that way when you're assuming things. And then, oh no, don't do that. Okay.
When to update?
And then I'm also going to open in my browser so I can. Okay, so this is the app. It's very nice. So I'm just going to go through the steps that I would. There are some instructions for people who want to run it. Also just instructions in terms of the visualizations, and also system requirements for you to run it. And then, so I'm going to upload that data file that I just showed you, which is this. I'm not going to use the example, but I did generate an example that is from the actual function, and then I added some noise. It's just like 12, and it's just like to get a good sense of things. So for the project title, I'm just going to say demo. And I told you it's 2 to 48 hours with a 2-hour resolution. Since these are unpaired replicates, which goes back to what you asked before, where essentially they're just clones of each other, so it really doesn't matter what their labels are. We also had 3 replicates, and we look for rhythms between 20 to 26. And I'm not going to smooth the data just because it takes a little bit longer. And I can run JTK cycle. I also want to remove unexpressed genes because I don't want those to factor in.
Is that the frequency? 20 to 26?
Yes, so like 2 pi over period. Yeah. So it'll be 2 pi over 20 to 2 pi over 26. Well, other way around.
That's your period in hours, right?
Yeah. Yes.
What?
What?
What over frequency?
Yeah. Well, but like when you transfer it to radians.
1 over 2 pi times frequency.
Yes. So I would click find rhythms if I actually wanted to sit here for like 10 minutes or so, but I don't want to. So I'm not going to do that. I already ran it. But then I would download the results using these buttons, basically the CSVs and my R data file, so we can look at some of the results from that. So
How much faster is it when you run it on the cluster?
I can't get it to run on the clusters.
No, but I thought we had it running.
I haven't actually run it on the cluster just because I ran into problems and it's hard to debug. So yeah, I don't know, but it's run in parallel, so it can use any amount of cores in there. So I would presume it runs a lot faster than my 4-core running 3 cores computer.
It ran. I ran it. It did definitely.
Yeah.
Half the time.
Yeah, which you would expect.
That's what I thought.
Which you would expect if you're running in parallel.
Well, we'd have to take it, we'd double check the parallelization and stuff like that.
Yeah.
But I just wanted to make sure people are aware this is running on the idea cluster also.
Oh, yeah.
She's running this locally.
And no parallel speed now.
Yeah.
The one thing I'm noticing is that a lot of these with the very high, very low period are also overdamped or overdriven.
Yeah.
Is that an artifact of the basically saying we're not looking at periods shorter or longer than that?
Yeah, probably. Because they just what?
No, go
just because they want to fit it as best as possible within their parameter space. One thing I would assume also happens is that it kind of gets stuck at that period. So imagine my starting point was maybe like 22 and it starts moving towards 20. And it can't really bounce back because it's in that local minima already. So maybe like 23 was better, but it started moving the other way. So I can presume that happens, getting caught in local minima like that.
Do you see that not happening as much when you just open it up to?
No, definitely not. Yeah, it doesn't happen. Just because it doesn't really get caught, it starts decreasing.
You know, it would be interesting would be to show several different things, partly for us who are trying to understand it, and more importantly, so one of the things that's really cool about running the data analytics against the biological data is sometimes you find things that biologists know are wrong, and that's when the really big thing. So when they tell you, we're only going to look at 20 to 26 because nothing interesting can happen outside there, that's when you should immediately say, let's try a few others.
Yeah. When we originally ran this data, we didn't actually impose those cutoffs, and we got things from like 14 to like 30, which we call like infraradian and ultraradian. And those are definitely interesting. The problem is just when you want to compare to JTK cycle, which can't do that. But I've also run JTK cycle with a longer range, and JTK cycle does end up missing things in a larger range. So it's just basically putting us on the same foot.
Yeah, Jim, the very first gene that we took to do the testing for the filters, just had a beautiful 16.5-hour period. Absolutely perfect. And hey, this is interesting. Well, no, it's not circadian though, so
Yeah.
go away.
Yeah.
I see. So the definition is that
The definition of circadian is close to 22.5. Basically.
So
push it back.
There's other biology involved. They've got their hands full with this biology.
So this is kind of what the output looks like for Echo, where we get all those parameters that I was talking about, also p-values, and also it puts the original data, and if you scroll, it puts the fitted data as well. We could also look at what JTK gives out in terms of a CSV, which is basically, this is what they give, essentially the period and the phase shift and the amplitude, and also p-values. I also added the Benjamini Uccelli just for accurate comparisons between us. So going back to the app.
So it only fits it on integer. Those are the reference.
Yeah, so it only does it. So it won't find things between things. So mine will. Mine will find decimal values. I know it's like kind of debated as well, because like if you sample at a rate of 2, you shouldn't find any more. He also finds less than 2, right? He just says 21, 22, 23, etc. And there is eJTK cycle, which I didn't really talk about, says, hey, we can't actually look at things like that because you didn't sample at that rate. But yeah, so it's kind of like debated, but I don't care, I guess. So
it sounds like you got like a million things better than going on. So
yeah. So we'll move on to the visualization part, which is pretty cool, I think, at least. So I'm going to upload that R data file that I ran earlier. I did run JTK cycle, so I'm going to check that. I'm not going to include overdamped or overdriven genes in my calculations for circadianness. So let's say I didn't actually really know what was going on. So I'm just going to look at a summary, a Venn diagram of the differences between these. And I get that plot that I showed you before. Yes, except I use the Benjamini Uccelli, so I'll change it back to the Benjamini. Yes. So yeah, same plot that was before. And we can see just in a text form what I showed you as well. It also gives you a confusion matrix, which basically says how we compare to JTK cycle. Did we both agree? Did they agree, etc. And so you can see all of those there as well. So I want to kind of explore my gene expressions, but I don't really know what's going on and what's the best. So what I'm going to do is actually look at gene lists. So I'm going to say things that were only identified by me, which is ours. I'm going to,
I love the diplomacy. Ours, theirs.
Exactly. It's just a nicer way of saying it.
Randex, just call it.
Yeah. So this is a gene list of things that I've identified. I'm going to sort it by p-value just because I want the best one possible. So let's look at that. So I'm just going to copy paste this.
but that is a 20-hour period.
Yeah, because it just stopped there. So I'm going to look at a gene expression graph.
So again, it's not driven. It's just gamma positive versus gamma negative.
Hey, we know that.
Okay.
Awesome. So yes, so this is what this gene looked like. I don't actually know what it is, but it does look pretty good for the most part. The shading is between the maxes of mins between the replicates. And as you can see, there's a lot of variability in the data itself, which you would expect. So if we look at, we can also see like summary right here of like what's about.
Could you actually explain a little bit of that? So that visually it's very powerful, but I just want to understand what you're showing with the
Yeah.
that envelope.
So this is the max and the mins of the 3 replicates.
Okay.
So it's, and then obviously the black line is the fit. We can kind of see that a little bit better here when we look at also with the replicates on.
Oh.
Yeah, so you can see, obviously it adds the replicates.
Those might have been, like, if those are scatter, because they're not strict replicates,
Mhm.
they're not matched, then you could get away with dots instead of.
Yeah. Another way of visualizing the data.
Well, let's look at the discontinuities in the lines.
That's missing data. Yeah. So, yep.
And what would happen, you've got 3 replicas here, so at 3 points, what would happen if you just took the replicate point closest to your fitted curve and made a new time series out of that and put it back in?
What do you mean? Sorry.
Well, I'm just saying, you've got the potential to make any kind of replicate you want, right? So
In terms of, did the biologist do or I do?
No, this is just purely from an operational thing here with your data. So you could make a new replicate by simply taking the one of the 3 points that's nearest your fitted line, right?
Yeah, I could do that.
And then run it again, and maybe you'd get a better fitted curve. Now, I don't know how legitimate it is.
Yeah, it might be biased,
but,
I think.
mean, because you're biasing it by the previous result.
Yeah.
On the other hand, you could look at it as maybe this is a way to converge on a better result. I don't know.
But then you'd have to run it twice, I think. But we
Another
kind
10 minutes.
And is this your
Yeah.
is this your algorithm's fitted curve
yeah, the black line.
applied to your data?
Yeah.
Do you think it might be better to just simulate many different just points that pretty much cover for like full better coverage and show like smooth and then like have your data colored around that curve just so it's
Yeah, you could do that as well. If you send that to me on Slack, I might implement it.
In other words, code it up.
Well volunteered.
And along the lines of what I was just talking about, I know Kristen proposed this where you just take the replicate, just scramble them up into 3 new replicates.
We did,
and
yeah.
do that several times and see how much difference you have.
Yeah.
in the fitted curve.
We tried that. It just takes a while, as you would
Because you've
expect.
got to do multiple times. But how much variation was there actually in the result? Was it significant?
So the person who implemented it ended up having some problems doing it. But there was a large, this was also when we didn't really have this inverse variance really in there at that point. So it's, who knows,
And
really?
were they filtering the data too?
No. They didn't smooth it. It was, yeah, but it's just kind of like, yes, you could probably get a more accurate p-value if you bootstrapped it, but it also would take you forever.
So you just
So,
throw
yeah.
more processors. John's going to buy us a whole other cluster.
Well, she's got to use up the ones we have.
I was gonna say, as soon as we started, I start seeing that the numbers are taking too long to run on our current cluster, which is using enough cores. Then we'll go talk to Chris Crothers and get her up to 6,000 or 8,000 cores. And then after that, then we'll start talking about real dollars. We have plenty of computation available, and frankly, this is something that everyone should be thinking about. Is we have a tendency to say, it's such a pain in the ass to move this to the cluster. I'm just going to run it on my thing because I'll still get my doctorate eventually. And by the way, when people suggest these really nifty, little things we could try that would take 10 minutes, we're like, well, it would take 2 hours. I don't have 2 hours or 2 weeks or whatever.
So what's very cool, and it's unfortunate, to be fair to Hannah, she really hasn't had, we only were able to actually get Echo running on the cluster like last week.
I'm just saying to everybody, it's really worth the time to get stuff on clusters because that extra, you pay the tax once and then you get the benefit lots of time.
And we have it set up on kind of uniquely the way idea. It's incredibly easy. It's literally copying your app into a particular folder And I mean, there were some things in Hannah's app. What's that?
I said the parallel and stuff.
Oh, that as well. Well, so there's the sort of the publishing it. So Hannah's app is actually published internally within RPI. So a biologist doesn't even have to have R. They can just go there and run it, which is very cool. And you've got the computes of the cluster. We run Microsoft R Open and with Microsoft kernel libraries. So certain things are automatic parallelization. Plus, if you've got certain things coded, which I believe the students did, the interns did, right? Didn't they do certain parallelization?
Oh, yeah. The
so-
reason that it runs as fast as it is very much thanks to Chendi, who's not here, but he was good about that.
So there's several different things that have gone into it, but the magic bullet, and you've actually been our test subject running it on the cluster, and you've seen the performance.
I can find everything. I will do everything.
You're the error finder. You're the alpha. So, anyways, maybe we're at the very end. We could actually just as an existence proof, we could go to the thing. Are you going to talk about the significance of you discovering the overdamped and underdamped?
Yeah.
I can talk about it afterwards after this demo, if you ask
Okay,
me about
do
it.
your demo. Do your demo. No, do your demo.
Yeah. Well, continuing.
Started at the campfire.
Well, so I also do know about white collar 1, which is a core clock gene in Neurospora. So I'm going to look about that, see how that is. It's 2, 3, 5, 6. Let's see how that looks. It looks pretty good. And as you can see, it's really damped. In fact, other methods have not been able to identify these core clock genes for a very long time. And it's probably because they are so damped. And yes, so this is just a cool thing. And so I also kind of want to look at, now that I've looked at a couple, I kind of want to look at how they look like for the entire data set. So I'm going to look at a heat map. They also tend to use this in their figures and stuff like that. So it's a little too big for the screen, but we sorted it by phase, and then we also normalized it to be between -1 and 1. And you can see there's kind of a wave of expression over time. So yeah, it's just a pretty cool thing.
How do you read this heat map?
Yeah, sorry. So gene labeling in image is very hard. So what it is, it's so each row is a expression and each column is a time point. So you have time point 1, time point 2, time point 3, etc. So
John McErick did this one.
Yeah.
That's perfect for his thing, and then we could get an interaction out of it.
So Hannah, yellow is the crest of the wave and blue is the trough.
Yes.
Just think of it coming out of the page.
So say that again. So time is going across.
Yep. And each row is a gene
And each row,
expression.
Okay.
Yes.
And you've ordered
So what does it mean that we see these? So
It's sorted just by phase.
Is this the -pi to pi? Is this been adjusted, or are we still having from 0 to 2 pi?
It's 0 to 2 pi. Yes.
But you've just ordered by phase shift, so
Mhm.
you get progressively more phase shift as you get down the plot.
Mhm. Yeah.
It's a good way to visualize the
Data.
circadian sequence.
Yeah. It's just they tend to use this visualization a lot in papers. So hence the reason why we did it. But it also looks cool. Also, April coded it originally, so props to April. So yes. So I also want to look at parameter density graphs. I want to see how they're distributed over everything. So I'm going to look at gamma, our forcing coefficient. And as you can see, I get a plot very similar to the one I showed you before. And we can also look at, I don't know, let's say p-values, maybe. Let's see that. And as you can see there, which you would kind of expect. Well, actually, there are so we have those cutoffs. Obviously, you cannot have p-values less than 0. And we said you can't have ones greater than 0.05. And you can kind of see that there are actually quite a lot clustered around less than 0.01 and less. So it seems like fits are either really, really good or they're just not good at all, which is kind of interesting, and I didn't know, I wasn't expecting when I chose this plot. So yes.
Does that say they're either circadian or they're not?
Yeah, well, it seems like the fits are either really good, and they're less likely to be less good. Does that make sense, I guess? Yeah. Okay. Well, that is the end of my demo. So questions?
So the question about the significance, because in a talk, I don't know whether it was this summer or at the beginning of the term, there was a discussion of how this underdamped and overdamped hadn't really been observed before.
Yeah.
And so there's a whole, where does that work? What do you think it means, and where's that going to go?
Yeah, so we actually kind of explored that in our conference paper that we published in August on this data, but using averages. And we found, I believe we found, like, don't like quote me on this because I might be wrong, but I think that, yeah, you can go find it on the internet, ACMVCB 17. But I think we found for damped genes that there were a lot that for their functional categories were related to, I think, it was either driven, I think, from metabolism. So a lot of them were involved in metabolism and like protein function and things like that. And you saw something very different for damped genes or vice versa. So it is something interesting. Why is it doing that? It is very interesting to look at things like that because you do learn more about the genes overall. So yeah, and one thing that they didn't even expect were driven genes. Driven genes was not a thing that they thought, because experimental conditions you would think, oh, definitely goes down over time. They also increase over time because of like stress response and things like that. So obviously they'll probably, if you took a long enough time course, they'd probably also decrease eventually. But for the small time period, they try to produce more in order to fix their loss of light and stuff like that. So it's just a lot more information for biologists to have, which is cool.
So I still have some questions about the replicate, or the replicate method and the smoothing. Can you pull up the first gene that you showed?
With the very first one?
Yeah.
Okay, I don't actually know what it was. So
Maybe, I think you had it in the slides too.
white collar
maybe
one?
Not, maybe not.
No, well.
Because I
Just find it in the app.
Yeah.
yeah, I'll just do this one because I. Yeah, so like this.
Right. Yeah. So actually, if you can show with the replicates.
Mm-hmm.
So in order to do this visualization, you have to upload the RDA file.
Mm-hmm.
So when you do smoothing, are you smoothing within the replicate and then across the replicates, or across the replicates and within?
So one thing that I would click like here, this unpaired, actually affects how we smooth things. So the fact that they're unpaired and it really doesn't matter does affect how we smooth it. We just use an average in order to smooth it. So we say we're going to take the average on this side and the average on this side to contribute to the middle point, because we can't really differentiate between labels, right, at that point. So yeah. Because
it looks like, so even just for purposes of visualization with that gene, it looks like you could just as easily, like for time point 3, you could swap blue and red, and you can basically say upper, lower, and basically kind of rank within there to show that. I was just worried that was actually affecting how the smoothing was occurring.
Yeah. Questions? Yeah, go ahead.
So, for the first one, the periodicity that you can observe in most of these plots for a specific gene, chances are it's going to be correlated to the function of some other protein within your cells. So, given that you're getting fairly good data from the method that you developed, do you think it's possible to be able to extract those type of relationship networks from the data set that you're generating?
Yes.
And my second question pertains to a comment you made at the beginning where you were basically excluding all of the genes that were expressed at less than 70% of the time. And I wonder, do you think you're losing any value by doing that? And I asked that in relation to my initial question because as something is, experiencing a particular cycle for a circadian rhythm, there's going to be some biological molecule that is going to either be driving it up or down. And in the graph that you had where those genes that were expressed less than 70% of the time, it seems that there was some periodicity there that might be out of phase work.
Yeah, exactly.
So those are my two questions.
Yeah. Okay. Let's see. Do I remember the first one? What did the first one have to do with? I actually don't remember, so you should say it again.
Relationship networks.
What? Oh, yeah. Yes. So you can actually take the results from this and look at networks. That's actually what we're going to do after this. After this is all wrapped up and done, we're going to look at tensor methods to do that, and basically take those groups from that, what we determined to be circadian, and try to look at networks between them and stuff like that. So we're definitely going to do that, but maybe not in a high throughput way. Who knows?
Doesn't matter as long as you get something.
Yeah. Yeah.
That's always a nice visualization, because that's what goes on the floor.
But yes.
The campfire.
And then in terms of your second question was, am I maybe missing 70% by like excluding those? Yeah. Maybe. That's why it's a checkbox. You can run it with and you can run it without. But I found, so 70% is definitely like a biological standard. It's not something like I made up. So it's just definitely getting, and I have never looked at a distribution of how many time points exist for a specific data set or anything like that. Maybe that would be enlightening. But for our another colleagues, Emily Collins, who also works on this, her mouse data set tends to have a lot where just they have one peak, which means basically nothing. So, yeah.
Can you go back to the heat map that you had?
Yeah.
And actually, the other thing though is that with low expression genes, that I mean, basically once you get to a certain point, you look at a distribution within a sample. There's basically it's bimodal. You have the expressed genes and you have the unexpressed genes. The unexpressed genes are basically noise.
No. So for this, do you think you can make it interactive so like the user can go to a particular region and look at it?
You could totally do that. It takes a lot longer to render. So I made all of these basically static. There's a package called Plotly that you can use to do interactive visualizations, but no, it just takes a really long time to render those. So yeah, but yes, you could do
There's probably like a lot of data points that are making this image, right?
Mhm.
So Plotly fails for like
Yeah,
using a lot of
but
data
there are
like
some
that.
solutions.
Yeah.
Like what?
Well,
I'm
I don't
curious.
know.
I'm curious. Because
I'll send
I
it
wanna
to the
know.
list tomorrow.
Okay.
But this is a known problem.
Yeah.
But dealing with it there are possible solutions to it.
Yeah, in fact, like I was dealing with this heat map earlier today, and you can't really use heat map, the actual function, just because this is too much data for it. So I use just image to visualize it instead.
What about the Plotly function
That takes even longer
that's what I
than
thought.
just a
That's
heat
what I
map.
But
So then it
so
would be interactive.
the other thing is
Yeah,
there are other alternatives also to how you render the images, well, actually, there are ways to render these as SVGs and throw them up, and that's a lot more efficient than putting the PNGs up.
Mhm.
So, so there are many different things, and those just ask me.
Yeah,
Maybe in the
SVG
dark
recs
picture,
are very
after
they're
I
surprisingly cheap.
Yeah, and that's actually something we're looking at with for the next phase for some of the other visualizations, such as the SWAT clock. All those are PNGs, and if we just went for SVG.
Oh, are you talking about in R?
The SWAT.
Oh.
I'm talking SWAT,
Okay, yeah, not like maps.
No.
Yeah.
We're not talking an artisanally crafted custom code for him.
Yeah.
So these are the things we have to try these things, see how they're broken, and then we can optimize it. But I was thinking exactly the same thing. Why didn't she drilling in on the individual genes using plot weight? The reason is because this is the mother of plots for plot weight.
Oh yeah, even just doing like 1,000 and just heat map 2 took a good like 10 minutes.
So for the uninitiated, Plotly is simply a way to take a visualization created by R like this and make it a little bit more interactive, have tooltips that allow you to fly over and see exactly what this particular gene is at what time. But the problem is there's an efficiency, there's a trade-off.
Yeah.
Using image, that probably was
Oh yeah, that's
instantaneous.
Yeah, but it's not
2
interactive.
cents.
That's the thing is it's not interactive. It's a pretty picture, but how do we go further?
Right,
Yeah.
We can drop that data set into all sorts of things, visualize.
Yeah.
Really nice to wrap that around. Say the circular core with information in it.
Well, yeah, right. So what we're talking about is so there's a couple of directions. One is other more visually impactful ways to render this, but the other is how to make this interactive. Because that's a conversation we have with the case people is how do we go further? How do we jump into it?
Yeah, but John, a lot of the stuff we have now, given it, you took it from a spreadsheet, turned it into an image, and are now doing your visualization against the image. Remember, there's a whole lot of other tools that are in the things in the spreadsheet to make it rendered and dragged, et cetera, in different ways, including ones we may have to build. Right? So again, we don't get hung up on whether it's a paint or an SVG or whatever, because that's what these things render, because they don't have the tools for going another way. Anything that says 10 minutes is a long time, sorry, I'm the biologists would giggle.
Oh yeah, no. I like technically running that
Because,
data set was not
running
very.
a small protein
Yeah.
expression only takes about 4 weeks.
Yeah.
So, it's almost
Yeah.
7. Was
Yeah.
there anything else that you wanted to?
There's nothing else I want to say in particular. But if any, I can take like probably one more question. Like if
What are the next steps in terms of?
Mhm.
So how general purpose this is, for example, how
So-
ready is it to turn over the biologist and have them beat up?
basically ready at this point. There are just like a couple like aesthetic things that I want to add, like a progress bar and things like more nice to use. But otherwise, in terms of functionality, it's done. And so we're starting to work on the paper to publish this. Hopefully done by the end of December. Definitely done by the end of December. But yes. So,
That was just recorded. Professor Bennett is getting this video in the morning.
Free announcement.
All right.
but yes.
So if there's no more
I
questions,
just have one other question. So you had to generate the R data file and then load it up, right?
Mhm.
Is there a way to make it seamless, like without having to download and then
You-
upload?
It's so yes, you could, but then yes, but I feel like it's not too much of a big deal to do it both like just to
Right.
download it and then upload
I'm
it.
just curious.
Yeah, so I feel like the trade-off is in terms of assuming that they're moving from the session it's worse than just saying, well, just upload it every time so that I could step away from this and then come back later without it running again and not have run the find rhythms tab and just straight up go to the visualization tab. So I think the trade-off, if that makes sense. Yeah, it's a little easier.
all right, let's thank Hannah.